So now let's start with the transduction. Let's say this is our bacteriophage, that is virus. This is our bacteriophage, that is virus which infects bacteria. It is a virus which can infect bacteria. As I have already discussed, in the viruses there is presence of two main components. One is nucleic acid. Yes, this is nucleic acid represented by double-stranded DNA. But for the simplicity, I have shown only single-stranded condition. Around this double-stranded DNA, this is proteinaceous covering. This is proteinaceous covering. This part of the covering, this is known as head. And this part of the covering, it is known as tail. This is head. And this one representing tail. Now this bacteriophage, it infects bacteria. Let's say this is our bacterial cell. Bacterial cell. In the bacterial cell, this is main DNA. Main DNA, which is double-stranded DNA for the simplicity, again showing single-stranded condition. Now this bacteriophage has infected the bacteria. Now what is this part of the bacteriophage? This part is tail. At the tip of tail, yes, at the tip of tail there is presence of enzymes. There is presence of enzyme, one enzyme which is associated here, that is lysozyme. This lysozyme has ability to digest the wall. Wall that is made up of the PG that is digested by the lysozyme. जैसे यहाँ पर wall digest होगी, there is formation of a hole. Yes, this virus will drill a hole in the cell wall. Now what will happen? The tail of virus will contract. Tail is going to contract, due to which now this double-stranded DNA, it is released inside the bacterial cytoplasm. So are you getting this? Lysozyme will digest the cell wall, due to which there is formation of a hole. Now this tail will contract and this double-stranded DNA of virus, it is released in the bacterial cytoplasm. So what is this? This is virus DNA and this one is representing bacterial DNA. Now this virus DNA, it will integrate itself in bacterial DNA. ये जो वायरस का डीएनए है, ये बैक्टीरियल डीएनए के अंदर इंटीग्रेट हो जाएगा, लाइक दिस. The virus DNA has shown the integration inside the bacterial DNA. When virus DNA is present in integrated form, then this is known as prophage or it is known as provirus. Yes, there should not be any confusion here. When the virus DNA is present in integrated form, then it is known as provirus. But when plasmid is present in the integrated form, then it is known as episome. So episome is for integrated plasmid, and this virus DNA is when integrated, then it is provirus condition. Now this virus DNA is in integrated form. जैसे बैक्टीरिया का डीएनए रेप्लिकेट करेगा, along with the bacterial DNA, there is also replication of viral DNA. जब तक वायरस का डीएनए बैक्टीरिया के डीएनए के साथ इंटीग्रेटेड है, this virus cannot harm the bacteria. In the integrated condition, virus cannot harm the bacteria. This type of the bacteria which contains provirus, it is known as lysogenic bacteria. It is known as lysogenic bacteria. The viruses which can show this type of the condition, they are known as temperate viruses. And this type of the life cycle of virus, it is known as lysogenic cycle. Are you getting this? Bacteria which contains provirus is known as lysogenic bacteria. The viruses which can be present in the integrated form, they are known as temperate viruses. And this type of the life cycle shown by the virus, it is known as lysogenic cycle. Now, generally this virus DNA, it remains in the integrated condition, but sometimes this virus DNA, it can excise out. It can come out. It can come out in two ways, that is symmetrical or asymmetrical manner. Sometimes this virus DNA, I am writing it like this, vDNA, it can come out from bacterial genetic material. So you can see here, जब वायरस का डीएनए बैक्टीरिया के डीएनए से बाहर आया है, तो इसके अंदर वायरस का डीएनए भी प्रेजेंट है, but along with this there is also presence of bacterial DNA. This type of the excision, जहां पर वायरस के साथ बैक्टीरिया का डीएनए भी बाहर आएगा, it is known as asymmetrical excision. Asymmetrical excision. वायरस के डीएनए के साथ साथ the bacterial DNA, it is also removed. Now this virus DNA, once it is out of the bacterial DNA, now this virus DNA, it will utilize the machinery of host. It will utilize the machinery of bacteria. By utilizing the machinery of bacteria, it will form one enzyme, that is nuclease. Nuclease is an enzyme which can digest DNA. Yes, this enzyme can digest DNA, and here it will digest bacterial DNA. This enzyme will digest bacterial DNA. Once the bacterial DNA is degraded, now this virus, by utilizing the machinery of bacteria, it will form its different body parts. I am repeating it here again. जब वायरस बैक्टीरिया के डीएनए से सेपरेट हुआ, वायरस का डीएनए, it will utilize the machinery of bacteria. and after utilizing the machinery forms enzyme that is nuclease. ये जो एंजाइम है ये बैक्टीरिया के डीएनए को डाइजेस्ट कर देगा. Genetic material of the bacteria is digested. Now what will happen? This virus DNA will form its different body parts by utilizing the machinery of host. By utilizing the machinery of host, now this virus DNA, it will form its different body parts, that is head, formation of head, formation of tail, head formation, formation of tail. Along with that, the virus DNA has also shown the duplication. But now, along with the virus DNA, there is also small fragment of bacterial DNA. Right? जब different body parts form हो गए, अब ये body parts assemble करेंगे to form mature virus. So now, the body parts, they will assemble. Now the body parts, they will join inside the bacterial cell. So this is head joined with the tail, right? And inside there is presence of viral DNA. But along with that, some fragment of the bacterial DNA is also present. Now it will cause the lysis of bacteria. Bacterial cell will undergo the lysis. Due to the lysis, now the viruses, they are released from bacteria. ये जो वायरस है, अब इस वायरस के अंदर अपना जेनेटिक मटेरियल प्रेजेंट है, but along with that it also has some fragment of the DNA of bacteria. This type of virus, which is having virus as well as bacterial DNA, it is known as transducing phage. Yes, this virus is known as transducing phage. Yes, what is the feature? Along with the bacterial DNA, there is also presence of virus DNA. Virus plus bacterial DNA. Now, such type of the viruses, such type of the cycle where the virus kills the bacteria, causes lysis, this type of the cycle, it is known as lytic cycle of bacteria. Are you getting this? When the virus, it kills the bacteria, result in its breakage, result in its lysis, this type of the cycle, this is known as lytic cycle. Now, this transducing phage is released from the bacteria, and this transducing phage, now it will affect another bacteria. Let's say this is a different bacteria. In this bacteria, there is presence of genetic material, main DNA, and transducing phage. Yes, this is transducing phage which has infected this bacteria. Now again the same procedure will happen. A hole will be drilled here. Now the tail will contract and this DNA, it is released here. DNA of the virus having the fragment of bacterial DNA. Now once the virus DNA is released here, now the next step, it will show the integration, right? So integration will occur. This one, virus DNA. Along with that, there is integration of bacterial DNA. So now you can see, ये जो बैक्टीरिया है, इस बैक्टीरिया का ये अपना जेनेटिक मटेरियल है. This is endogenote. And this white DNA, it belongs to different bacteria. It is transferred in this bacteria with the courtesy of viruses. तो ये जो DNA है, ये किसी और बैक्टीरिया को belong करता है. This is exogenote. Endo and exogenote together, what is going to represent? Going to represent merozygote or partial zygote condition. Right? So here you can see in the bacterial cell, genetic recombination has occurred. This transfer of DNA from other bacteria to this bacteria. And here it is assisted with the help of viruses. So this whole mechanism of transduction is very much clear to all of you. So this is all about the sexual reproduction. Now I will be starting with the different bacterial groups. First of all, I will be dealing with the cyanobacteria. Now I will be starting with the bacterial group, that is cyanobacteria. In the two-kingdom classification system which was given by the Linnaeus, cyanobacteria was placed under algae in the class that is Mixophyceae. Placed under the class that is Mixophyceae. Mixo means slimy covering. Around the cyanobacteria there is presence of mucilaginous slimy covering, and phyceae, this suffix we know it is utilized for algal class. This group is also known as cyanophyceae because this algae is blue in color, that's why known as cyanophyceae, also known as microalga. Microalga, it is also known as BGA, that is blue-green algae. It is largest gram-negative bacteria. I am writing gram-negative like this. It is also known as oxyphotobacteria. Now, why it is known as oxyphotobacteria, that we have to see here. This is only bacterial group which contains both photosystem 1 as well as photosystem 2. ये सिर्फ एक बैक्टीरियल ग्रुप है जिसमें photosystem 1 भी है, photosystem 2 भी present है. Now the photosystem 2, if it is present, it means it is involved in the water breakdown. If there is water breakdown, means there is release of oxygen. तो जब ये बैक्टीरिया photosynthesis perform करेगा, now there is release of oxygen, so we can say it performs oxygenic photosynthesis. Oxygenic photosynthesis. Due to the oxygenic photosynthesis performed by cyanobacteria, primitive earth atmosphere, which was anaerobic, that was changed to present day earth atmosphere, that is aerobic. Are you getting this? This is only bacterial group which perform oxygenic photosynthesis. Due to the oxygenic photosynthesis, जो primitive earth का environment anaerobic था, due to the release of the oxygen, it turned into present day aerobic environment. Right? So the oxygen which is added in the environment for the first time, it was added due to the activity, oxygenic photosynthesis of cyanobacteria. Now, cyanobacteria, first we will see with respect to their occurrence. First we will see the occurrence of cyanobacteria. Now, the cyanobacteria is cosmopolitan in nature, that is present almost everywhere. Cosmopolitan in nature. Cyanobacteria can even be present in stress conditions like frozen lakes, hot water springs. Frozen lakes, as well as hot water springs. So it means in the stress condition like hot water spring, there can be presence of both archaebacteria, which we have discussed previously, and it can also be, there can also be the presence of cyanobacteria. Both can be present in the stress condition. This is only bacterial group which shows association with almost every eukaryotic group. Shows association with almost every eukaryotic group. तो प्रोकैरियोटिक ऑर्गेनिज्म है, पर ये एसोसिएशन शो करता है with every eukaryotic group. So I will be citing few examples here. One, it shows association with fern, aquatic fern it is, that is azolla. Shows association with aquatic fern, that is azolla. In the azolla, it is associated with leaves. Leaves के साथ associated है, and the cyanobacteria which is associated with the leaf, that is anabina. Yes, this is associated with the leaves. Also shows association with gymnosperm, that is cycas. Cycas, this is our gymnosperm. In the cycas, the association of cyanobacteria is in the roots. Roots showing the association of cyanobacteria, they are known as coralloid roots. Very important, right? Now the cyanobacteria which is associated with coralloid roots, that's again species of anabina. Right. Now after the occurrence, now I will be dealing with the body structure of cyanobacteria. Now the body structure. Cyanobacteria can be unicellular or multicellular. I am writing one example for unicellular cyanobacteria, that is Anacystis. Multicellular cyanobacteria can be colonial or can be filamentous. One of the colonial cyanobacteria is microcystis. So there should not be any confusion. Microcystis is cyanobacteria, and you will study one name, that is macrocystis. Macrocystis is largest size brown algae, and brown algae is a eukaryotic algae. Are you getting this? Microcystis is prokaryotic alga, and macrocystis is largest brown alga, that is eukaryotic. In the filamentous, example is nostoc, oscillatoria, and anabina. Now, what filament is going to represent in the cyanobacteria? In the filament, this type of cellular organization is present. This type of cellular organization, cells, they are placed one above the other. This structure, it is known as trichome. Around the trichome, there is presence of slimy covering, characteristic feature of myxophyceae. Myxo means slimy. इसके आसपास ये present होगी slimy layer. Right? तो ये हो गया trichome. इसके आसपास slimy layer. Two of them together will represent filament. Right? So this is all about the cyanobacteria. So in this lecture, we have studied reproduction in the bacteria. Along with that, I started with the cyanobacteria, dealing with the occurrence and body structure. So in the next class, I will be dealing with the rest of the part of cyanobacteria. Thank you so much.